In this video we will discuss about the two different kind of clusters that we have seen in our previous video, that is Tumor-T cells clustered and APC-T-cell clustered. So far we have understood in our previous video a small difference between clustered cells, clustered T cells and the singlet T cells. That is, the clustered T cells has more tumor reactivity. That is, the antigens that are formed in these clustered T cells are more specific to the tumors to which they are interacting, compared to the singlet T cells, which has more reactivity towards virus. And this is just a general standard or general norms for the bystander T cells. So here we will discuss in this video to one small topic. It is, what are the difference that researchers able to find out between these two different kind of T cells clustered. That is, T cells that are interacted with tumors and the T cells that are interacted with the APC. The tumor T cells, this APC T cells, what they are able to see that the T cells has more exhaustion and co-modulations compared to the T cells that are clustered with the tumors. And these things are shown in this panel F figure, where we can see that exhaustion in this T cells from the APC clusters are higher compared to the T cells from the tumor case. And these things they have validated they have also shown this using two examples, that is CD28 signaling and CTLA4 signaling. We can see that they are high when they are high in the T cells which are clustered with the APC. And thereafter they also did this arrow transparency studies using their TCR-seq data. So in this they have used the data that they have generated using Tcellreceptor seq, and they check the receptor ligand interactions. And what they are able to see is that the interactions of the receptors that are commonly found in these exhaustion T cells are more with the APC compared to the tumors. And there are some also non-specific interactions that they mark them as gray color. So this further validate the findings that they have seen from this TCR clonotype. So in this case, in the first figure of this panel F, they checked the most expanded TCR clone, most expanded or top 30 most expanded TCR clones that they are able to find. In that case, they are able to see more exhaustions in the T cells that they observed from the APC cluster. And these things they validated using the TCR or the Tcellreceptor seq data. And other things also that they are able to see that the co-modulations. Yeah, so co-modulations is not specifically mentioned in this figure, but it is mentioned in the text that the co-modulations interactions between the T cells and the APCs are better, and that will lead to the more expansions of this exhausted T cells or T cells exhaustions when the APCs are interacted with the T cells. And based on these findings, they are able to relate these findings. Yeah, based on this finding, they are able to again conclude or make an inference that it is commonly known that the T cells that are recovered from the tumors can be reinvigorated and again activated to act against the tumors. And this thing is not true for the T cells that are recovered from the APC. So that point further validates the findings that is observed in this studies, that the more exhaustion cells are found when they are clustered with the APCs, and that is one of the reasons that it's difficult to reinvigorate to use those T cells against the tumors, as they will act in the reverse order, and they will suppress the T cells response, whereas the T cells that are recovered from the tumors can be reinvigorated or re-energized to act against the tumors. So I hope you understood the difference between tumor T cells and APC-T cells cluster. I might have repeated some things multiple times so that everything is, or most of the things are cleared to you or to the audience. That's all for this video. If you need to understand more about this topic in detail, you can go to the reference that has been provided in the description. Thank you for your time.